Awesome. So, hello everybody. My name is Jorik Andeweg, and I'm going to be presenting on my final project that I did for Dr. Gumbart's biophysics class in the fall of 2019. And I did have a partner with this project. His name is Prahlad Iyengar. I think he's an electrical engineering major, so he didn't help me with this presentation or anything, but he did help me with the original project. This project was a computational reproduction, or a simulation, of a historical experiment in which the two strands of a DNA molecule were mechanically separated. So this presentation is divided into three sections. First, I'm going to talk a little bit about DNA background information and its physical structure. Then I'm going to talk about the original experiment performed by Bockelmann et al. In 1997. And finally, I'm going to talk about my computational reproduction of this experiment. So DNA consists of two strands, two polymer strands that are wound around each other in a double helix shape, giving the appearance of a twisted ladder. And the two strands are held together by hydrogen bonds, which can be separated by a modest force because hydrogen bonds aren't true chemical bonds, they're more like weak attractions. Each DNA strand consists of nucleotides strung together, and each nucleotide has two parts of interest to us: the phosphate backbone, which makes up the side of the DNA molecule, and the nitrogenous bases, which jut inwards into to form the rungs of the ladder, and they bond to each other to form those hydrogen bonds that I talked about. So the sequence of nucleotides in the DNA molecule carries information that's important to cell processes. Most importantly, the sequence of amino acids that it needs to form proteins, which regulate all cell processes. And there are different complementary base pair combinations. So there are four types of nucleotides: adenosine, which is represented by an A, thymine, which is represented by a T, cytosine, C, and guanine, G. And if you see a particular base pair on one strand, you know what the base pair across from it on the other strand is going to be because they have specific pairs. An A always bonds with a T, and they connect with two hydrogen bonds, as shown in the image on the very right side. And a C always pairs with a G, and they form three hydrogen bonds. As a result, the C-G pairs are generally a little bit harder to separate than the AT pairs because they have more hydrogen bonds. But if you want to know the exact energy that it takes to separate it, you have to refer to this table, because it depends not only on whether a given base pair is an AT pair or a CG pair, but also on what the next base pair down the sequence is. So here we see the hydrogen bond, the delta G of hydrogen bond formation, the change in Gibbs free energy, for a base pair at index I. And it depends not only on the base pair at index I, but also on the base pair at index I plus 1, so the next one down the DNA sequence. And as you can see, the delta Gs are generally larger in absolute value in the CG area than in the AT area. These values come from John Santalucia. He found them in 1998. The final DNA property that I want to talk about is that when you have a single DNA strand, which is also relevant to the experiment, those are quite flexible. So you can imagine that it sort of likes to get messy, like a shoelace is rarely just naturally straight. If you apply a force to stretch it out, so you try to separate the two endpoints of a single strand of DNA, then you might imagine that it's energetically favored for it to elongate and reach its sort of maximal conformational length. But it's entropically favored to bunch up because there are more conformations available to it if the end-to-end distance is smaller. So Stephen Smith et al. In 1996 found the following relationship between the force you apply to a DNA molecule, a single-stranded DNA molecule, and its end-to-end distance. So here, the X in angle brackets is the average end-to-end distance or extension of the DNA molecule. It is a statistical process, so sometimes it'll be shorter and sometimes it'll be longer, but this is the average. L is the contour length of the DNA. F is the force that you apply. Kb is the Boltzmann constant, and T is the temperature of the surrounding area. And this relationship comes from a combination of theory and experiment. It comes from a statistical analysis of a polymer model called the freely jointed chain model. And with these particular values, it was curve fit to best fit DNA. And it turns out to be a pretty good approximation. Now I'm going to talk about the original Bockelmann et al. Experiment. It was performed by these people in 1997, and the goal of it was to mechanically separate the two strands of a DNA molecule by pulling them apart at a constant speed and measuring the force required to do so. So they used a particular DNA molecule with a length of 16 millimeters and a known sequence of about 50,000 base pairs. They used this variable x0 to represent the position of the force lever, or the separation of the force lever from the other end of the DNA strand that they were separating, and they steadily increased it at 20 nanometers per second. And they used this force lever to measure the force. So it obeys Hooke's law. It's a good approximation. It's bendable, so it kind of behaves like a spring, with a spring constant of 1.7 piconewtons per micrometer. So by optically observing the deflection of this force lever, they were able to tell the force required to separate the DNA strands. And here are the results. So on the x-axis, we have the displacement, or the position of the force lever. And keep in mind that this was steadily increased over time, so you can also think of the x-axis as representing time. And on the y, we have force, the force required to separate the two strands. And there's two features in these results that I want to talk about. Firstly, just the general magnitude. You'll notice that it takes about 12 to 13 piconewtons to separate the two DNA strands from each other. And secondly, Bockelmann et al. Noticed what they called a sawtooth pattern. So that's characterized by a gradually rising force followed by a dramatically dropping force. Gradually rising, dramatically dropping, gradually rising, dramatically dropping. So that indicates stick-slip motion because there are two alternating periods of behavior. First, not a lot of links are opening up, not a lot of separation is happening. So that means that the force is going to steadily build up in the force lever as the displacement increases. Then, when the force reaches some critical value, that's followed by a rapid period of a lot of opening of DNA links, so a lot of separation in rapid succession, and that causes the force lever to relax and the force to dramatically fall in a short time. That's stick-slip motion. And finally, I'm going to talk about my computational reproduction of this experiment. So we used the same DNA sequence that Bockelmann et al. Used. We found it on GenBank. And we did not do a full molecular dynamics simulation or anything like that. That would be way too computationally expensive. So you might have been imagining like a 3D simulation with NAMD or a similar software, but that's not what we did. Instead, we took kind of the opposite approach and had a very minimalistic simulation. So we described the entire state of the system with only two variables. Firstly, x0, the position of the force lever, and secondly, n, which represents the number of open links. In other words, the index of the fork in the DNA sequence. We set both initially to 0 and then iteratively updated them. So to update x0, all we had to do was increment it a little bit, since x0 was, in the real experiment, increased at a steady speed over time. And we incremented it by a very small amount with each iteration, much smaller than the length of a single nucleotide. And to update n, with each iteration, we would give it three options. We would give it a choice. It would either go up by 1, symbolizing that a link opened, it would go down by 1, symbolizing that a link closed, or it would stay the same, symbolizing that there's no change in the system. And this is called a Monte Carlo simulation because any simulation in which a system has evolved over time in an iterative and random way is called a Monte Carlo simulation. So updating n is governed by Boltzmann statistics. Some of you may remember from statistical mechanics that the probability of a system to occupy a particular state is proportional to the Boltzmann factor, which increases with decreasing energy. So if a state takes a lot of energy to occupy, it's not very likely that it will be occupied. But since it's on a small scale and there's a lot of thermal fluctuations, there's still a chance. So our goal here is to find the energy E of the system as a function of n and x, which are our two state variables. And if we can do that, then we can find the probability of n opening versus closing a link. There are three energy terms we need to take into account. The hydrogen bond energy, and this we can read right off the table supplied by Santa Lucia, since we know the sequence of the DNA molecule, and we also know the current position of the fork. We know the current n value. So we can read right off how much energy it would take to form a new bond or break an existing bond. Then we have the force lever, which, remember, behaves like a spring, so it has potential energy associated with it. We know from classical mechanics this is 1/2 kx squared. And finally, the single DNA strand, which, since this also has its own force extension relationship that we've already talked about, supplied by Stevens, this also has a potential energy associated with it. So to find the potential energy associated with the force lever and the single strand of DNA, the first step is to find which force will result in the correct total extension. you can see from the diagram that the total extension of both the single strand of DNA and the force lever together equals x naught. So for a given x naught value, using the force extension relationships of the force lever and the single DNA strand, we can find the force responsible for that extension. And then if we know the force and we know the distance, then we can integrate force over distance to find potential energy for the single DNA strand. And since the Stevens equation is quite complicated, we had to do this numerically. Here are our results. So again, it's displacement on the x-axis and force on the y-axis. And you can see that they're very similar to Bockelmann's results. We had a force in sort of a similar range, about 13 piconewtons. And we also saw the sawtooth pattern indicative of stick-slip motion, where we had a gradually rising force followed by a rapidly falling force. Here are the two results side by side, so you can kind of see the comparison. The scales on the axes are just a little bit different. So the two large peaks that you can see over here in my results are about the same size as these smaller peaks in Bockelmann's results. But again, I was pretty pleased with these results overall. They were very similar. And here finally is an alternate way of representing our results. So now we have the number of open links, n, on the y-axis, and still displacement, x naught, on the x-axis. So you can see that there were periods of relative stasis where n wasn't changing a whole lot, followed by periods of rapid link opening where tons of hydrogen bonds were being destroyed and the DNA was zipping open quite a bit in rapid succession. So those are the more vertical sections of the graph where n is rapidly rising. I have a few final remarks. Some of you might wonder, why do we care about DNA's physical properties, particularly the way that it separates? And it turns out that DNA separation is something that occurs quite a bit in the cell. So whenever the cell needs to read the DNA sequence to make a protein or it needs to replicate its DNA, it has to separate the two DNA strands. So by doing experiments like this and learning about the force required to do that, we can learn a little bit about the cell's energy budget, for example. What would I change if I were to do this project again? Well, when I implemented this simulation originally, I implemented it entirely in Python, which is a high-level language, and I ran it on a virtual machine, which does not have low-level access to my computational resources. So it was very slow and inefficient code. Since then, I've learned a lot of good coding practices, and I think I'd be able to make it much more efficient if I ran it, if I created it again. And finally, I want to talk about the fact that this project demonstrates the usefulness of simulations as microscopes. So when Bockelmann originally interpreted his sawtooth results as being stick-slip motion, that was more of an inference. And by computationally reproducing this experiment, we can sort of confirm that his suspicion about the cause of the sawtooth pattern is correct. Because if you look again at this alternate representation of our results, we can see that there are indeed alternating periods of static n versus rapidly increasing n. So this sort of confirms Bockelmann's interpretation of stick-slip motion. And it also confirms that these results that Bockelmann experienced arise fully from first principles that we understand, and that there's not some underlying factor that we don't understand, because if that was the case, then I wouldn't have been able to reproduce the results with my simulation. And finally, are there any questions from you guys?
Thank you. We have time for maybe one or two questions here. So members of the audience, why don't you, if you have a question, unmute yourself and ask it. Or you could raise your hand. How about that, Martino? Go ahead.
I'm wondering if there's anything expected that would be slightly different if you go with a three-dimensional simulation in the future, even though you said it was computationally expensive, I guess.
Right, so that would actually be orders of magnitude too computationally expensive. If you look at this graph again, we opened thousands of base pairs, and each base pair contains maybe dozens of atoms along the length of the nucleotide of the DNA molecule. So simulating thousands of base pairs worth of DNA is completely unrealistic. But I suspect that if in the far future we develop the resources to be able to pull off that kind of simulation with full quantum mechanical atom interactions, I suspect that the results would be very similar to what we did here.
All right, thank you.
There was a question in the chat.
I can't see the chat. Would you mind reading it?
Says, was this from your biophysics project?
Yes, this was my final project for Dr. Gumbart's biophysics class. A few of you may have seen it before if you were also in that class, although this is a new presentation.
So I have a question for you. So this is a statistical phenomenon. If you repeat this, do the sudden openings occur in the same locations on the DNA? Are those like soft spots of the DNA, or are they random?
Yes, that's a good question. There is, in fact, a pattern that emerges, which is that the stick sections, the periods where the force will gradually increase without any new links opening, are more likely to appear in the CG dense regions of the DNA. And I actually wanted to include a graph showing that, but I decided that there wasn't enough time to. But it's very cool. If you show a graph of the CG content of the DNA as a function of position, as a function of nucleotide like index, and then overlay a graph of force versus index of position along the DNA, then you'll see that it is in fact the periods, the sections with a lot of CG bonds, which are hard to separate, where the force will have to build up to a higher value before any links start separating.
All right. Thank you very much. Thanks a lot. I think that's all. That's all for you. And so we will. I think maybe I will momentarily stop recording, because that might help me separate these two things from each other. I will momentarily stop recording. Matt, you're on deck. If you could switch over, take control of things, and be ready to present, I'm going to just stop and then restart recording, and then we'll move on to you.
Thank you.